Alright, this video is your pre-lab video for your PCR crime lab. Okay, and what we have right here is the CFTR protein. It is a transport channel protein that is found in lung tissue, on lung epithelial tissues exactly. And what it does is allows chloride ion passage out of the cells into the mucus that coats the epithelial cells. Now, what happens if you have a faulty CFTR gene is it is possible for you to get a disease called cystic fibrosis, which is a debilitating and fatal disease. And the reason why I'm bringing this up is because this is the star of the show in terms of this crime lab. And the reason why is because it is a whodunit, so to speak, about a missing woman named Missy Baker. So I'm going to go ahead and give you guys the opportunity to read this. So by all means, pause the video, read through it, and then I'll continue on. So what we have Missy Baker. She's missing. There are two suspects. Pretty much one of them, or both of them, have a blonde hair or a hair that is in their vehicle that they believe to be Missy Baker's. So what we are going to do is we are going to identify if the hair in their car contains the CFTR mutation. And if it does, then we know that Missy Baker was in that car, because unfortunately Missy Baker does have cystic fibrosis or has that mutation. So looking here, just kind of give an idea as to exactly what it does. So here's your CFTR protein right here. Okay, these two little lobes right here. And they act as a channel to allow chloride ions out into the mucus. And then because of the high chloride concentration, that's going to result in osmosis. So water comes down in here, resulting in the hydration of the exterior of the epithelial cells. Unfortunately, if you do not have the chloride channel open, so in this case with somebody who has a CFTR mutation, the mucus unfortunately becomes viscous and sticky, and it's going to trap a whole bunch of bacteria and viral particles and toxins that could result in definite respiratory diseases, but also death. So unfortunately for Missy Baker, she has this disease, but fortunately for Missy Baker, it is very, very rare. So the rarity of this disease will actually help us identify if she was in one of those two cars. And spoiler alert, she was in one of those two cars. So let's kind of take a look at a gel electrophoresis. In this case, we have a 100 base pair ladder, which is the same type of ladder you guys are going to use in class. And then we have H and D. The H is representing a healthy CFTR gene. And so according to this right here, you could estimate that a healthy CFTR gene is approximately 825 to 850, somewhere around there, base pairs long. And for Missy Baker, she has an approximal 400 base pair deletion in that CFTR gene. So in the D column, where would you expect to find, or the D lane, where would you expect to find the band for somebody who has a deletion like Missy Baker? Right? Well, hopefully we can do math, and we realize that about 400 or 850 minus 400 is about 450. So I don't have a white pen, so I'm just going to use a red one. And so we would expect to see a band right about there, okay, for an individual that has that deletion. Okay? Maybe green will show up a little better. All right, none of them are really good, but you know where it is. It's right here. Okay? So here is our experiment. So you're going to have 4 tubes. Tubes A and B are going to be representing the hair DNA samples found in A and car A, suspect A's car and suspect B's car. And then you're going to have 2 controls. You're going to have a healthy control, right? So a healthy CFTR gene, which is longer than the deletion or the deleted CFTR gene that's going to be found in tube D. So you're going to have 2 controls and 2 experiments, in this case A and B. So what goes into a PCR experiment? Okay, my recommendation is if you don't remember about PCR, right, and why it's relevant, then I would recommend going to the Utah Genetics website or just go into Google and type in PCR Virtual Lab Utah Genetics, and that should be the very first thing pop up. Do that first hit and then go through that lab. You are going to do that lab. In fact, I would recommend you do the virtual lab as a refresher. So do that now before you even continue with the rest of this video. So come back if you need to. So now that you are back, the template DNA that you're going to need to amplify, okay, amplify means to make many, many, many copies of it, is going to be needed, which is going to be suspect A, B, H, and D. Okay, so A, B, H, and D are going to have your DNA template. You're going to have your DNA primers, which are where the DNA polymerase is going to attach at specific regions of the template DNA. You'll have DNA polymerase, you're going to have some nucleotides, so A's, T's, C's, and G's, and you're going to have a buffer that contains magnesium ions as well as its ability to maintain your pH during this reaction. Now these 3, as mentioned right here, this is called a master mix. So all 3 of those things are in the master mix solution in the appropriate amounts. So you're going to have 3 components that you're adding to your tubes. Okay? And we'll get to those here in a second. But your process of thermocycling, which is PCR, revolves around 3 concepts. 3 steps. One is denaturation of the DNA, one is the attachment of primers, and then copying of the DNA. So just so we know, denaturation is going to happen at 94 degrees Celsius, which is hot, really hot. Annealing, which is the attachment of the primers, so the temperature cools down, and then the temperature rises up a little bit to 72 degrees Celsius, and that will allow DNA copying to be made. Now, if you're wondering, well, those are really, really, really high temperatures, because 72 degrees Celsius, 37 degrees Celsius is body temperature, right? 98.6 degrees Fahrenheit. So 72 degrees Celsius is way up there. I want to say it's somewhere around 160 degrees Fahrenheit, okay? And you're thinking, well, how can this enzyme work so well at such high temperature? And the reason why is because the DNA polymerase that we're using actually comes from a certain organism that lives in hot springs. So it's normal, 161 degrees Fahrenheit or 72 degrees Celsius isn't anything. That's kickback. Right, that's nice and relaxing temperature for it, so it's able to do its job. Ours is 36 or 37 degrees Celsius. Its is 72 degrees Celsius. So that's why we use a very high temperature polymerase that came from a bacteria that can survive and flourish at really warm temperatures, since PCR requires really hot temperatures. So moving on, right? This is what's going to go into your tubes. You're going to have 4 tubes, 1, 2, 3, 4, right? And in each tube, you're going to have 15 microliters of your master mix, which is the buffer, the magnesium ions, the TAC, which is the DNA polymerase. Okay, TAC DNA is the DNA polymerase, and then your A's, T's, C's, and G's, and your DNTPs, right? You're going to have 10 microliters of the CrimeLab primer mix, so these are your reverse and forward primers for the PCR process. And then you're going to have 5 microliters of the suspect template DNA. Okay, so in there you're going to have 20 microliters of solution. Okay, so that will go into the PCR, the mini PCR machine, which is this guy right here. Okay, a little clear, about the size of a tissue box, and he's going to be connected via USB to a computer. In fact, there's going to be 2 of them paired up to each computer. Now, I'm going to go over how to set all this kind of stuff up, but you are going to actually enter all this stuff manually. So I'm going to go over the parameters, and hopefully after watching the review and watching this video and understanding what PCR means, then you'll get what I'm getting at. So the initial denaturation is going to happen at 94 degrees Celsius, and it's going to take 30 seconds. And then 5 seconds longer is going to start the denaturation cycle. And then you're going to have the annealing cycle for 5 seconds at 57 degrees Celsius, and then you're going to have the extension at 72 degrees Celsius for 8 seconds. So if you remember, this is denaturation, DNA breaking apart. Annealing, that's when the RNA or the DNA primers attach. And then extension is when DNA polymerase is going to make the copies of the DNA. Now, these 18 seconds right here are going to be repeated 30 times because every single time that it goes through a cycle, we have an exponential growth of DNA. So, or at least of the fragment that we want. And then lastly, we'll have one final extension of to pretty much tie up any loose ends at 72 degrees Celsius for 30 seconds. Okay? So, one thing I want to mention, I'll mention in class, but I want to mention it here too, that this, right, we're approaching 200 degrees right there, 200 degrees Fahrenheit. Here, 160. This, about 140. So, I mean, hot. So, understand that the computer is going to say the lid is hot, so don't touch it, okay, because it's mainly the lid part that's going to get hot because that's where the heat block is in the thermocycler. So be careful you don't burn yourself on the thermocycler, okay? So be mindful of that. All right, if you guys have any questions, shoot me an email or leave a comment in the classroom.